Hello, everyone. I thought we would talk about the latest episode of The Twilight Zone, which is our life. Let's listen to this message from our president.
Hi, perhaps you recognize me. It's your favorite president. And I'm standing in front of the Oval Office at the White House, which is always an exciting place to be. I got back a day ago from Walter Reed Medical Center. I spent 4 days there and didn't have to. I could have stayed at the White House, but the doctor said, because you're president, let's do it. I said, fine. You tell me what to do, and I'm going to listen. These are great professionals. They've done a fantastic job. And by the way, not only at Walter Reed, all over the country, we have the greatest doctors in the world. We have the greatest nurses, the greatest first responders, law enforcement, by the way, incredible. Firefighters, everybody. They're just great. We have great people. This is a great country.
Just great.
But I spent 4 days there, and I went in. I wasn't feeling so hot. And within a very short period of time, they gave me Regeneron. It's called Regeneron. And other things too, but I think this was the key. But they gave me Regeneron.
Yes, he just said that 3 times. So let's look and see. What is this Regeneron? We have, it's in September 29th, Regeneron today announced the first data from a descriptive analysis of a seamless phase 1, 2, and 3 trial of its investigational antibody cocktail, RNAi, showing it reduced viral load and the time to alleviate symptoms in non-hospitalized patients with COVID-19. RNAi showed positive results, positive trends in reducing medical visits. So ask yourself, why on earth would they give the sitting president of the United States an experimental drug that has not even completed its phase 3 trials? I don't buy it. I mean, I think that would be terribly risky, and it's just completely insane to me. Here it says October 2nd. This is on Wikipedia on us from about RNAi. Says October 2nd, Regeneron Pharmaceuticals announced that President Trump had received a single 8-gram dose of RNAi after testing positive for SARS coronavirus 2. The drug was provided by the company in response to a compassionate use temporary authorization for use from the president's physicians. Compassionate use? Isn't that when someone is critically ill? Did he look like he was in dire straits when he got on that helicopter to you? He did not to me. This is compassionate use is the use of an unapproved drug or medical device under special forms of investigational new drug applications outside a clinical trial by people with serious or life-threatening conditions who do not meet the enrollment criteria for the clinical trial and process. So, do you think he was in a life-threatening situation? Well, I don't know. I would argue that that's a bold-faced lie. But let's go on and listen to what our president has to say.
And it was like unbelievable. I felt good immediately. I felt as good 3 days ago as I do now. So I just want to say we have Regeneron. We have a very similar drug from Eli Lilly.
Let me pause that because I'm going to jump over here and show you. Let's see. I have to scroll down to find Eli Lilly. Okay, this article, I apologize, from BioCentury, DARPA's gambles might have created the best hopes for stopping COVID-19. A U.S. defense agency that specializes in turning science fantasies into realities jump-started technologies and nurtured companies that are now at the forefront of the response to the COVID-19 pandemic. The Defense Research Advanced Projects Agency, or DARPA, has taken risks where others wouldn't. Its pursuit of high-risk, high-reward technologies, combined with its mission-driven approach to managing projects, is promising to pay off in the fight against COVID-19. DARPA was behind the creation of the DNA and RNA vaccines, funding the R&D by Moderna Inc. and Inovio Pharmaceuticals. Let's scroll on down here. We'll come back to the RNA and DNA vaccines. Let's talk about Regeneron, the fact that they're using antibody. Although RNA and DNA-based vaccines could be a major advance, there are inherent disadvantages for the use of vaccines in general pandemic response. The problem with vaccines is twofold. One is that oftentimes you don't know a lot about your pathogen, so it can take a long time to know which components on the surface of that pathogen would be the right component to inject. Even in the case of COVID-19, where the spike protein target was rapidly identified because of prior research on the related coronavirus SARS and MERS, yielding safe and effective vaccines in less than 12 to 18 months is not feasible. The other disadvantage, Jenkins added, is that it can take weeks or months after a vaccine is administered for it to produce immunity. In some cases, 2 or more shots are required. Antibody-based therapies could offer a faster route, an option DARPA and companies like Regeneron Pharmaceuticals and Vir Biotechnology are pursuing. We'll come back to this Vir Biotechnology. Unlike vaccines, these monoclonal antibodies can provide immunity rapidly to yield therapeutic and temporary prophylactic effects. There is precedent for the use of monoclonal antibodies to prevent infection. I can't even pronounce this drug name. Was approved by the FDA to prevent serious lower respiratory tract disease caused by RSV in pediatric patients at high risk. 2 monoclonal antibodies are being used to treat Ebola patients in the Democratic Republic of Congo, one from Regeneron and another discovered by Humabs Biomed in South Africa, developed by the NIH's National Institute of Allergy and Infectious Diseases. Humabs, which was acquired in 2017 by Vir, was a subcontractor on the DARPA Ebola antibody discovery contract. I can't help but think of Mel when she writes Ebola. She calls it Ebolize. Anyways, one of the past barriers to use of antibodies in pandemics was that it took up to 2 years to discover an effective antibody. DARPA and other government philanthropic funders and industry have invested in technologies that have cut the time required to screen for and discover antibodies to days. In 2018, DARPA launched the Pandemic Prevention Platform that seeks to develop a scalable, adaptable, rapid response platform capable of producing relevant numbers of doses against any known or previously unknown infectious threat within 60 days of identification of such a threat. As part of P3, DARPA awarded a 4-year, $30 million contract in 2018 to AbCellera to apply its antibody discovery platform to viral pandemics. That's almost 3 times the 10.8 million in venture funding AbCellera had raised at that time. AbCellera, like Regeneron and Vir, initiated an antibody discovery program when the sequence of the virus that causes COVID-19 was announced. The company received a blood sample from one of the first patients in the U.S. known to have recovered from COVID-19, and within a week, identified hundreds of antibodies that could be used to create a product with therapeutic or protective properties. AbCellera, which is using DARPA funding for its COVID-19 research and development, has signed an agreement with Eli Lilly Company for co-development of an antibody-based product to protect against and or treat COVID-19. The company expects to select antibodies, manufacture them, complete preclinical tests, and start a phase 1 trial within 4 months. See AbCellera, Vir, find partners for COVID-19 mAb monoclonal antibodies manufacturing capacity. AbCellera also has partners with Gilead Sciences, Inc., Novartis AG, Sanofi, Pfizer, and Teva Pharmaceuticals. Ambitious goals for P3. We'll come back to their pandemic preparation. Let's see, where is that? Pandemic prevention platform in a moment. But let's go back and finish listening to what the president says.
And they're coming out, and we're trying to get them on an emergency basis. We've authorized it. I've authorized it. And if you're in the hospital and you're feeling really bad, I think we're going to work it so that you get them, and you're going to get them free. And especially if you're a senior, we're going to get you in there quick. We have hundreds of thousands of doses that are just about ready. I have emergency use authorization all set, and we got to get it signed now. And you're going to get better. You're going to get better really fast. This is things that nobody even thought of a few months ago. The job that the scientists, the labs, everybody have done is incredible. Then in addition to that, you have various other drugs that help a lot. But I view these, I know they call them therapeutic, but to me, it wasn't therapeutic. It just made me better. Okay? I call that a cure. So I want to get these things done. So we have to get them done. We have to get them approved. And I want to get them to the hospitals where people are feeling badly. It's much more important to me than the vaccine. But on the vaccines, we have many companies that are in final stages for the vaccines. Johnson & Johnson, Moderna, Pfizer, all great companies, but many of them. And we're going to have a great vaccine very, very shortly. I think we should have it before the election. But frankly, the politics gets involved, and that's okay. They want to play their games. It's going to be right after the election. But we did it. Nobody else. Nobody else would have been able to do it. The FDA has acted as quickly as they've ever.
The fucking dumbasses.
Acted in history. There's never been a time, and no president's ever pushed them like I pushed them either, to be honest with you. But the FDA is approving things in a matter of weeks that used to take a matter of years. So we have these drugs, Eli Lilly, and the others that are so good, but they are, in my opinion, remember this, they're going to say that they're therapeutic, and I guess they are therapeutic. Some people don't know how to define therapeutic. I view it different. It's a cure. For me, I walked in, I didn't feel good. A short 24 hours later, I was feeling great. I wanted to get out of the hospital. And that's what I want for everybody. I want everybody to be given the same treatment as your president, because I feel great. I feel like perfect. So I think this was a blessing from God that I caught it. This was a blessing in disguise.
Blessing from God. You hear that?
I caught it. I heard about this drug. I said, let me take it. It was my suggestion. I said, let me take it. And it was incredible the way it worked. Incredible. And I think if I didn't catch it, we'd be looking at that like a number of other drugs. But it really did a fantastic job. I want to get for you what I got, and I'm going to make it free. You're not going to pay for it. It wasn't your fault that this happened. It was China's fault. And China's going to pay a big price, what they've done to this country. China's going to pay.
China and Anthony Fauci with this $3.7 million collaboration with the Wuhan lab after the gain of function was stopped in the United States. Okay.
Pay a big price what they've done to the world. This was China's fault. And just remember that. So we're going to get you the drug. It's going to be free. We're going to get it into the hospitals as soon as you can, as soon as we can. And you'll see some amazing things happen because we have our military is doing the distribution. It's called logistics. And they deliver hundreds of thousands of troops in a matter of days. This is easy stuff for them.
Hundreds of thousands of troops within days to deliver this.
Our general's all ready. We're waiting for the emergency use authorization, and the drug companies have just made a lot of it. So hopefully this is going to be not just a therapeutic, it's going to be much more than a therapeutic. You're going to get better, you're going to get better fast, just like I did. So again, a blessing in disguise. Good luck.
Good luck. You're going to need
perhaps
it.
You reckon
You're going to need it. It's certainly going to be much more than a therapeutic. he said it's logistics. They're going to roll out hundreds of thousands of troops are going to come out to roll out with Operation Warp Speed. In another interview, I recently heard him say that Operation Warp Speed was going to be greater than the Manhattan Project. In case you don't know, the Manhattan Project was a research and development undertaking during World War II that supposedly produced the first nuclear weapons. What else did we get from World War II? We got the Operation Paperclip, where over 1,600 scientists were brought over, such as Werner von Braun, to create NASA and what we have that passes for science today. So let's talk about these monoclonal antibodies. Let's see if we can go here. We'll talk about this. After months of incredibly hard work by our talented team, we're extremely gratified to see that Regeneron's antibody cocktail, RAIN-CoV-2, I can't help but see the word RAIN, REIGN, when I see that. It rapidly reduced viral load and associated symptoms, said George Yankopoulos. I don't even know if I'm saying that right, but if you remember, I talked about him in my Biochester video where we talked about Westchester County's 11 biotechnology companies, Regeneron being one of them in the forefront. So the thing is, let's see over here. This article, what is the antibody cocktail Trump received? It's a combination of two antibodies directed against a key protein of the virus that causes COVID-19, SARS-CoV-2. They bind to a region on the main surface spike protein that helps the virus attach to a receptor on human cells called angiotensin converting enzyme 2, or ACE2. The targeted region is dubbed the receptor binding domain. One antibody comes from a human who had recovered from a SARS-CoV-2 infection. A B cell that makes the antibody was harvested from a person's blood, and the genes for the immune protein isolated and copied. The other antibody is from a mouse, which was engineered to have a human immune system that had the spike protein injected into it. Oh boy, I feel better, don't you? There was an article that came out where they were talking about Regeneron using fetal cells. And they say they do not use fetal cells in this. What they were, they're using humanized mice. But I feel comforted. Do you? Anyways, I will put the links to some of these other articles down below. And there is some discussion that Trump possibly had stocks, had made money from stocks from this company. And whether that's true or not, I do not know. I do know that I saw an interview with the blonde woman who usually speaks for the president, and I can't remember her name, was saying how he had donated his salary to HHS this last month. So let's go on and talk about, this was from September 17th. It's called A DARPA-Funded Implantable Biochip to Detect COVID-19 Could Hit Markets by 2021. So we're going to skip over or move on from the monoclonal antibodies for a moment to talk about this. The most significant scientific discovery since gravity has been hiding in plain sight for nearly a decade, and its destructive potential to humanity is so enormous that the biggest war machine on the planet immediately deployed its vast resources to possess and control it, financing its research and development through agencies like the National Institutes of Health, the Defense Advanced Research Projects Agency, and HHS's BARDA. The revolutionary breakthrough came to a Canadian scientist named Derek Rossi in 2010, purely by accident. The now retired Harvard professor claimed in an interview with National Post that he found a way to reprogram the molecules that carry the genetic instructions for cell development in the human body, not to mention all biological life forms. These molecules are called messenger ribonucleic acid, or mRNA, and the newfound ability to rewrite those instructions or produce any kind of cell within a biological organism has radically changed the course of Western medicine and science, even if no one has really noticed yet. Rossi himself puts it, the real important discovery here was you could now use the messenger RNA, and if you got it into the cells, then you could get the messenger RNA to express any protein in the cells, and this was the big thing. Now, Moderna is working with DARPA, and Moderna has the messenger RNA vaccine, 1273. So we also have companies creating short interfering RNA vaccines. They're using short interfering RNAs. I was not familiar with that, so I looked that up and found that both the messenger RNA and the short interfering, or sometimes small interfering, or sometimes silent interfering RNA is part of the RNA interference. Now, when I see this acronym, I immediately see RAIN. My brain scrambles that around. This RNA interference, or RAIN, is post-transcriptional gene silencing. This has to do with gene expression, gene silencing. And both the messenger RNA and the short interfering RNA are part of this pathway. This is about genetically engineering, genetically modifying humans. But we were on the topic of monoclonal antibodies. So let's see. What is monoclonal antibodies? Okay. I know if I skip down here to Britannica, it says right in this little bit, they're produced artificially through genetic engineering and related techniques. There are laboratory-made versions of proteins naturally produced by the immune system in response to invading viruses or other pathogens. Neutralizing antibodies, whether natural or monoclonal, can bind directly to portions of viruses that they attach to and enter cells, preventing them from initiating the infection cycle. So they're claiming this therapeutic, which targets the ACE2 receptor. They bind to the ACE2 receptor. So I'm not exactly clear on what it is. The targeted region is dubbed the receptor binding domain. So I'm not sure what it is they are hoping to accomplish with this antibody cocktail, and what it is. But we have the National Institute of Allergy and Infectious Diseases involved in this. All part of this COVID-19 prevention network. SARS-CoV is the virus that causes coronavirus. Yes, interestingly enough, they have not isolated the coronavirus, this supposed novel coronavirus, COVID-19. They have not isolated this virus. In fact, we have this link. I watched a video by High Impact TV earlier today, about an 8-minute video, where he goes into the fact that this is now on the CDC or website, where they literally admit that no quantified virus isolates to the 2019 novel coronavirus are currently available. Assays designed for detection of the quantified, designed for the detection of the 2019 novel coronavirus RNA were tested with characterized stocks of in vitro transcribed full-length RNA. Now, interesting, because when I look down here, I noticed that these RNA mini kits, the real-time PCR assays, were performed using the Thermo Fisher Scientific path. So I had recently saw this link when I was looking into the RNA interference and what exactly the short interfering RNA were. I came across the fact that Thermo Fisher has silencer select short interfering RNA libraries. These are the latest improvements in short interfering RNA design, off-target effect prediction algorithms, and chemical modifications to yield short interfering RNAs with unrivaled efficacy, potency, and specificity. Sorry. In short interfering RNA screening applications, the result is fewer false negatives due to poor silencing and fewer false positives due to off-target effects. Silencer select short interfering RNAs yield cleaner, more consistent data from primary screen, reducing time, resources to find true, validated targets. As a result, there are fewer failed experiments due to poor silencing and cleaner, more consistent phenotypic data. Now, I find all that really interesting, because these short interfering RNA, well, here we have a simplified model for the RNA interference pathway. It says it's based on 2 steps, each involving a ribonuclease enzyme. In the first step, the trigger RNA, either a double-stranded RNA or a microRNA primary transcript, is processed into a short interfering RNA by the RNA's 2 enzymes, dicer and drosha. The second step, the short interfering RNAs are loaded into the effector complex, RNA-induced silencing complex, or RISC. They love their acronyms. The short interfering RNAs unwound during the RISC assembly, and the single-stranded RNA hybridizes with the messenger RNA target. Gene silencing is a result of nucleolytic degradation of the targeted messenger RNA by the RNA's H enzyme argonate, or slicer. If the short interfering RNA mRNA duplex contains mismatches, the messenger RNA is not cleaved. Rather, gene silencing is a result of translational inhibition. So all of this makes me think of as in the days of Noah, and how it talks about Noah was pure in his gene ration, or pure in his generation. And he was building the ark to prepare for the coming rain, the flood. And it looks like we're about to have some rain. We have this RNA interference, and we have them coming out and calling rain CoV-2. But all of this is rather fascinating when you consider the fact that they've never isolated the virus, so we have no idea what these PCR assays are detecting. We've heard all of this talk about the fact that there are false positives, when what it boils down to is this is all based on a lie. It's all based on fraud. Yet you have all of these companies coming out with all of these different RNA technologies, and they are going to save us, to prevent and or treat and or cure this deadly COVID-19 coronavirus. And I would just stress that I mean, this is literally to rewrite us on a genetic level and upload us to the cloud. Now, I'm going to bring your attention. Let's see if I can find. Okay, let me see the Mercola X. I'll put this link down below where Dr. Carrie Medej talks about these vaccines and the fact that they want to transform us, transhuman, transfection. Let's see, here we go. Well, that's not it either. Okay. Transfection. Now, the word transfection in the center here says it is a portmanteau of trans and infection. Genetic material such as supercoiled plasmid DNA or short interfering RNA constructs or even proteins such as antibodies may be transfected. Antibodies like the monoclonal antibodies that are already genetically modified, or the messenger RNA, which is CRISPR-Cas9. When I was looking in to the short interfering RNA pathway, I came across a thesis written in 2019 discussing building a lipid nanoparticle as a delivery system for the short interfering RNA. I'll read a little bit to you. The field of RNA therapeutics is currently undergoing both transformation and expansion. Specifically, research in lipid nanoparticle-based RNA therapeutics is gaining significant traction. Other research into mechanisms of gene regulation and manipulation, including short interfering RNA and the CRISPR-Cas9 system, have demonstrated the potential of RNA-based disease treatment. This work identifies a delivery system which can regulate expression of green fluorescent protein in human embryonic kidney cells, or the HEK293, stably expressing the green fluorescent protein. Analysis of the short interfering RNA-induced gene knockdown demonstrates that the current short interfering RNA lipid nanoparticle formulation is equally as effective as a commercially available transfection reagent, the lipofectamine, or RNAi, which is designed specifically for plate-based transfection experiments. The short interfering RNA lipid nanoparticle formulation can reduce the green fluorescent protein expression to 30% with cytotoxicity slightly lower than the RNAi. CRISPR formulations also resulted in the significant lockdown, though further research is needed to optimize and verify loss of green fluorescent protein expression. So, the RNA therapeutics are a rapidly growing field of pharmaceutics that have enormous potential for the treatment of hereditary genetic diseases and various cancers. Do we all have a hereditary genetic disease that we need to have rewritten? Because that's basically what they are planning to do with these new vaccines. In the United States alone, close to 1,800,000 new cancer cases are expected by the end of 2019, and over 600,000 are predicted to lose their battle by the end of 2019. Both the short interfering RNA and the CRISPR-Cas9 system have been successfully used in vivo to inhibit the expression of genes and for gene knockout. What gene is it exactly that they're planning on knocking out in us? And the goal of treating genetic disease and genetic underpinnings of cancer. Let's see. Accordingly, my project sought to develop novel strategies for the delivery of 2 gene therapeutics. To complete this study, a short interfering RNA delivery vehicle was developed by utilizing lipid nanoparticles. A novel delivery vehicle for the CRISPR-Cas9 gene editing system was developed, and 3 HEK293 were transfected to investigate the efficacy and cytotoxicity of the developed formulations. This project provides valuable information on the development of systemic RNA therapeutics and helps bridge the gap between the basic science and its translation to medicine. Now, this RNA interference basically allows them to regulate gene expression through manipulation of the target of endogenous messenger RNA molecules. Now, we'll go on down here to the, I'm going to scroll down to the bottom to talk about their conclusion. So basically, they transfected the HEK293 cells to express a green fluorescent protein. And with this lipid nanoparticle delivery system, they were able to deliver in the CRISPR guide RNA and Cas9 messenger RNA to, I'm sorry, hold on. Let me get down here to the, okay. All right. So they transfected the HEK293 cells to express the green fluorescent protein. So through the lipid nanoparticle delivery system, they inserted the short interfering RNA to knock down the green fluorescent protein. So it says the short interfering RNA lipid nanoparticle formulations produced in this study were successful in knocking down the green fluorescent protein in the HEK293 cells. These formulations had significant green fluorescent protein knockdown, low cytotoxicity, and could be empirically formulated to produce particles of low size and PDI. CRISPR guide RNA, messenger RNA, and guide RNA plus messenger RNA were formulated to produce lipid nanoparticles low in particle size and PDI. Lipid nanoparticle delivery of CRISPR guide RNA and Cas9 messenger RNA to target green fluorescent protein in HEK293 cells demonstrates that lipid nanoparticles may be a suitable vehicle for the CRISPR-Cas9 system. So basically, they were saying that it worked, and I just threw that in there as an interesting part. It's just something that I learned when I was looking into what exactly these short interfering RNA were. And as I said, they're sometimes called small interfering, sometimes known as short interfering, sometimes silencing RNA. But it's a class of double-stranded RNA, non-coding RNA molecules, typically 20 to 27 base pairs in length, similar to microRNA, and operating within the RNA interference pathway. It interferes with the expression of specific genes and complementary nucleotide sequences by degrading the messenger RNA after transcription, preventing translation. So, all of these technologies, including the antibody therapeutics, the antibodies which were genetically modified and transfected, this is all about transforming you and uploading us all to the cloud, connecting us to the AI, the beast system. Trump is not our savior by no means. He is part of the. Well, first of all, he's an actor. It's all theater. War is theater, and we are at war. The idea that he is going to save us and help us to get our constitutional republic back for the people, a government for the people by the people, is complete horseshit. He's going right along with the World Economic Forum. All of this insanity should have been stopped already. Even they come out on their own website, the CDC, and admits that they never isolated the virus. And so these draconian measures that they have taken, the lockdowns, the forcing us all to wear masks, to protect us from the dreaded COVID-19 disease, it's all fiction. It's theater, and it infuriates me. Because the majority of people have just gone right along with everything, and they don't even put up a fight. We have these public-private partnerships with these biotechnology companies, and DARPA and BARDA and the NIH and the AID. Funding this stuff. This is the government. This is big government. And they've been planning this for a long time. Event 201, and prior to that, Clade X, and with their planned pandemic response, the P3, I can't even remember what the 3 Ps are. P3 aims specifically, like I said, to develop a scalable, adaptable, rapid response platform capable of producing relevant number of doses against any known or previously unknown infection within 60 days of identification of such a threat. Well, I'd say they're past their 60 days. But there is no end in sight of this. And now with this, Trump coming out, supposedly having gone to the hospital and given these experimental drugs, and now he's saying that he's cured. He's telling people he's cured, and he wants everyone else to get this too. But let me just make a point. I was discussing making this video with Patrick, and he wanted to make sure that I brought up these points in the video. So let me just read them to you. Number 1, an antigen is a foreign protein, as is found on bacteria or virus, to which the body mounts an immune response with antibodies. 2, these freakass witches spliced human genes into mice so that the mice could make antibodies to whatever they injected into them, and then they harvested the antibodies to put in a needle. Number 3, Trump made millions from his shares in Regeneron that he invested in long before he started pimping out his shit. Another point to make, though, is I don't really think it's about the money. Trump just recently donated his salary to HHS. So, I hear people who are Trump supporters, and they just think it's the greatest thing because he's not getting paid. Well, I mean, he is getting paid. He may not be taking the salary as a president, but he's certainly in the pockets of the people running, the shadow government, the people truly running what basically amounts to a corporation, the United States, that our individual rights were gone long ago. We've had an illusion of freedom, which they are certainly now cracking down on. The World Health Organization is actually calling for more lockdowns, all based on a fraudulent test. A virus so deadly you don't know you have it until you have a test. And then what is it that they're testing? But number 5, when you shoot anything into the human body, it's seen as a threat. An immune response is mounted to that threat. So if you shoot an antibody into somebody that's supposed to protect you from a biological threat, you will make antibodies to the antibodies. This will guarantee autoimmunity and could literally kill you. The very thing that's supposed to be protecting you could kill you. And go back to C number 1. So, I am not fooled by any of this, the show that is being put on for us, and it really concerns me because people seem to have an inability to think for themselves, and likely because they're calling it a therapeutic. People will line up to do this. They will line up to take this and have no idea what it's going to do. Now, Trump is saying he's cured. What was this, a week ago? He took this, the MABs, the monoclonal antibodies, and now he's saying he's cured. What did their fake test now say? He's no longer testing positive. I don't know. But I keep saying that this is all to upload us to the cloud. And I want to show you a short little video I'm gonna link about this and the idea that let's see, that we're coming up with smart vaccines. Let me see. The quantum dot, connecting the vaccines. And I didn't even go into the ACE receptor, which is what these antibodies are targeting. And when I look at the Malachard's website and all the things that are affected by the angiotensin converting enzyme, or ACE2, there is 1,066 hits. And the Malachard's is a human disease database. So there's 1,066 hits for things that are affected by this ACE2. So that's horribly frightening to me. So I'm going to go over here and I'm going to show you a predictive programming from a cartoon on Netflix, Stretch Armstrong. So let's just watch a little bit.
Charter City is stunned by the sudden rollout of Rook Unlimited's most fantastic device ever. The Smart Mark. Throw your other devices away, because the Smart Mark is more than just a phone. It's part of you. Use it as cash. Book your travel. And even shoot video. Distribution points are all over the city. And don't let the lines discourage you. The Rook Unlimited Smart Mark is worth the wait. I never realized how wonderful technology could make me feel. The Smart Mark has brought order to my life. And the best part? It's absolutely free. A gift of thanks to our city from Rook Unlimited.
23% of the population has been given the Smart Mark, number 1.
Time to test our system, number 6. The Flex Fighters are out there somewhere. Teaming up with Jonathan Rook. Let's put our new recruits to work. Subtly.
We should find the Flex Fighters.
I ought to find the Flex Fighters.
We should find the Flex Fighters. We should find the Flex Fighters.
Growing up in this city, the streets of Old Town, I saw the worst mankind had to offer. Chaos, disorder. I vowed to make it all change. To make it all better. All these years working in secret. Rising up the ranks of the tech men. It was all leading to this day. Order has finally been established in Charter City. No child will have to fear for their safety again. The people owe you the greatest of debts, number 1. For liberating them from the chaos of their lives.
You guys love new tech. You didn't want to get smart marked?
No way. That's clearly some shady biz. Who wants to be...
You call yourself a man of science. World order is our destiny. Submit to a smart mark and join the glorious victory of order over chaos, or you will be eliminated.
Grandpa, Mr. Savick, where is everybody?
They all went to get those foolish new devices.
What? No, they can't. Grandpa, those things are dangerous. It's a conspiracy. Mind control.
I knew it!
3 hours is enough time for us to get to our families. Maybe we can stop them before they get marked.
They seem to be establishing checkpoints of some kind. This is how they keep order.
This is the nightmare I warned you about. People think smart marks are brand new root tech that will improve their lives. They don't know they're being tricked.
All smart marks so far. But your chip makes it possible to control everyone in Charter City, whether they're marked or not.
Now, whether these things are going to work like they are purported to, we don't know. And whether it's antibodies touted as a therapeutic or whether it's a vaccine, it comes in a little clear vial, and we have no idea. We didn't make it. We didn't watch them make it. And we are supposed to just trust them and let them inject this into our bodies. We have been taught not to question these witch doctors. They have all the answers and they know, and we shouldn't bother our pretty little heads with that information. We should just do as we're told. I will put all these links down below, guys. I'd like to hear what you have to say about all of this. Are you going to be pure in your gene ration?